Hi everyone, welcome back to another video tutorial. In this tutorial, I would like to walk you through a Jupyter script that will allow you to analyze your DeepLabCut CSV outputs. However, in this script, you are able to draw multiple regions of interest with different colors, the same way that I'm showing you here on the screen. So let's go through this really quickly, and then we will discuss some of the parts that you should be aware of and some of the changes that you should make to the script in order for it to get to work for you. So before continuing, I'm going to copy over the DeepLabCut script into the path that I have here. So now that I have that done, let's start and run each of these columns. So in the draw necessary ROIs, you have the ability now to draw different regions of interest, but make sure that you name them first. In my case, I'm using only 3 regions of interest. It is a T-MazeAnalysis in my case, so I'm using middle arm, right arm, and left arm. And the color designations for each of the ROIs is red, green, and blue. You could expand on this and do multiple more ROIs if you like. For my analysis part, this is just sufficient. So when I press run on this one, it gives me a T-Maze outline and then allows me to draw on it, but you have to make sure that you are drawing your ROIs in this order that you specify up here in the ROI names. So in this case, it would be middle arm first. So that's my middle arm. Then it would be right arm. So the right arm that how we designate the right arm is going to be on the right side of the middle arm, like so. And then same thing for the left arm. Like that. And then to fix these in place, you just press stop interaction, and that will also record the coordinates of these ROIs. Moving on to calculating time spent in each region of interest. Be mindful that my script is utilizing paired body part system. What this means is that 2 body parts, in my case thorax and head, have to be inside of the region of interest simultaneously before any entry, exit, or time spent can be calculated. Okay, so once I run this part of the code, it gives me the number of frames that the mouse has spent inside of each of the region of interest. Moving on to analyzing the movements, entries, and exits. In this part of the code, I'm adding in a debounce frame. What this means is that the mouse has to at least spend 15 frames within a region of interest before entry or exit can be counted. So in other words, if a mouse enters a certain region of interest, it has to have entered that region of interest for at least 15 consecutive frames, and that parameter can be changed here based on your own experimental needs. For example, if you're recording at 30 frames per second and you want to have a threshold that is 1 second long for your entries and exits, you can change this parameter to be 30 to reflect 1 second of your video. In my case, half a second is perfectly fine, so I'm going to leave this as 15. Right, so be mindful of this parameter as well as the parameter that I have defined as the paired body part. So in addition to debounce frames, the condition is that the head and the thorax have to be inside of the region of interest before the entry and exit can be counted. Moving on to generating visualizations for the entries and exits. This part, feel free to manipulate and change however you like. For me, the bar graphs work just well, and in this case it's showing me that the left arm had 9 entries, right arm had 9, middle arm had 19, and that the first entry ROI was the left arm. Let's move on to graphing the velocities. Please make sure that you replace frame rate here with whatever frame rate that you're working with. All of the outputs are going to be within pixels per second units, so make sure that you put in the correct frame rate here so that the conversion to seconds can be done properly. In order to visualize paired body part average velocities in each of the different regions, this code here can be quite useful. Moving on to tracking the sequence. For the T-MazeAnalysis, it's very important that we track the sequence in which the mouse is moving into different regions. For example, in this case you can see it's middle arm, right arm, and middle arm again, left arm, middle arm, and right arm. So what this signifies is that the mouse is moving from left arm to the right arm through the middle arm. Right, so all of these sequences for us become important in analyzing the T-Maze data, as these patterns indicate to us the curiosity of the mouse or tendency of the mouse to want to explore different regions based on different stimuli that we put into different arms of the T-Maze. So I'm going to run this, and this gives me a list like this. You can also utilize a CSV export function, so that you can export all of this data into a CSV file and save it wherever you like. The next parts are all temporal analysis, so to speak, and these are all done through graphing. So I have notes in here in my code for you to read and see what each part of the code is doing. And this is the last part of the code. This allows us to understand the number of entries over time bins. In my case, I've chosen to do 90 bins. You could very easily change this to 10 if you like, or whatever works for you. And this allows you to very quickly visualize the number of entries the mouse is making towards each ROI over a given time. For example, in the later half of the graph, this is towards the tail end of my experiment, so I can see that towards the tail end, middle arm entries are relatively stable, whereas the left and the right arm entries decrease. So it's a quick visualization to view the pattern of mouse's behavior over time. And for some people this can be quite useful. In my case, it's very useful because it allows me an visual indication of the pattern and frequency of mouse's entry into different body parts, different ROIs. Right, well, that's it for this video. Thank you so much for watching.